Okay, so again, I'll try to answer these questions as best as I can. We'll do our Q&A at the end of this content block and at the end of the lecture as well, and we'll kind of come back to some of those things that you guys were talking about. If anything pops up regarding specific questions, again, pop it in the Q&A. And if anything comes up regarding content that I go through, if you need me to explain it a little bit more, or you're kind of missing something, pop it in the Q&A and we'll cover it. So to start off, again, we're kind of easing in a little bit nicer here. We're starting off with cell structure and function. So we're looking at prokaryotes and eukaryotes. Again, these terms should be relatively familiar. It does depend on your school and obviously what you guys do learn in junior science. But generally things that I've highlighted here, like you'll kind of know if these are a big term. And I would start now making your glossary. And as you learn stuff in class, make that glossary and add to it throughout the year. So when you come to exam time and when you're learning how to do your practice questions, those definitions for these keywords, such as prokaryotes, eukaryotes, it's really clear in your mind. Okay, so in terms of some differences, so we can see our prokaryotes on the left and our eukaryotes on the right. Generally, and I want to say generally, because don't think of it strictly like this, but I like to use examples when I think of things in my head. So often I think of bacteria as our typical example of a prokaryote, and like an animal cell, for example, as our typical example for a eukaryote. Bearing in mind, there are more prokaryotes than just bacteria cells, and there are more eukaryotic cells than just animal cells. But for me, that's how I would kind of like remember it in my head. Like instead of thinking, what's the difference between a prokaryote and a eukaryote? I used to think in my head, what's the difference between a bacteria cell and an animal cell? But obviously, think about exceptions and stuff like that. Okay, so no membrane-bound nucleus in a prokaryote, and in a eukaryote there is, so that's what we can see kind of here. The general rule of thumb with these two is prokaryotes are very simple, eukaryotes are a lot more complex. So no organelles, we have membrane-bound organelles. We'll talk about organelles in a bit more detail in a second. But yeah, again, really simple, doesn't really need the technical stuff that organelles do, whereas eukaryotes do require it. Not capable of true multicellularity. So we technically think of prokaryotes, definitely in the context of Bio 1-2 at least, as this idea of single-celled organisms. Eukaryotes, we tend to think like a little bit more as multicellular, but be aware there are single-cell eukaryotes. So kind of maybe put this as like a difference that's a bit lower down your list in terms of ones that jump to your mind. Prokaryotes, we'll talk about binary fission a bit later on, but they reproduce by this like really simple method of dividing, whereas eukaryotes, we're thinking about mitosis, and for this actual idea of reproduction, we're thinking of meiosis as well, but we'll talk about that a little bit later. So as you can see, there's some kind of differences, and you can even see like visually as well, that idea of the nucleus is a big one, the organelles are a big one as well. Okay. So that's the stuff we can kind of see there. So organelles in general, here we have an animal cell. We've got a couple of organelles here. Obviously, you can tell by the way they're drawn. We've got like our endoplasmic reticulum and our Golgi, our nucleus. Some of the other ones are a bit more ambiguous. Basically, with your organelles, know what I don't want to say popular, but the major ones are, especially in like an animal cell or a eukaryotic cell. Know their general functions. You don't have to be too specific. Again, your teachers will emphasize which organelles you need to know heavily. On the study design, they emphasize chloroplasts and mitochondria. So those are 1000% you need to know them. But yeah, be aware there are some others where you just need to know their functions a bit more generally, and kind of what they may generally look like. Whereas, for example, chloroplasts and mitochondria, we look at them a lot more closely. But yeah, just generally know some of their functions, and know also what they will look like on a micrograph. This is a skill obviously needed in 1, 2, and 3, 4. So here we have kind of like our typical drawing of a mitochondria, and you'll know what they look like in a diagram as well, but understand what they will look like on a micrograph. When I say micrograph, like a picture of a cell taken with a microscope. So usually they're like, if you go through a couple of year 12 exams, you'll see kind of what I'm talking about. So they'll be like black and white, they'll be grainy, and they won't be this kind of clear, nice, color-coded, really distinct kind of organelle that you see like on images like this. So just make sure that you're looking at key features, because you may be asked in an exam to like label a micrograph. And if you're used to seeing mitochondria as this typical orange squiggle, and then you're seeing this in black and white and it looks a little bit different, then yeah, you may get a little bit confused. So kind of understand what it looks like, I guess, in real life as well. Okay, so here is a general table. Again, I'm not going to describe them in a lot of detail. Essentially, our lecture is 2.5 hours, so I will have to move through some things relatively quickly. Again, if you have any specific questions, pop them in the Q&A and I can come back if we have time. But yeah, because you guys are getting copies of these slides as well, just bear that in mind. So if we think of our really important ones, again, mitochondria and chloroplast, we'll talk about in a second. But we think about them, basically they are important and they're singled out on the study design because of their role in our biochemical pathways. So you guys would have likely heard of cellular respiration. So we think about that being associated with our mitochondria and our photosynthesis being involved with our chloroplast. Bear in mind as well that the details of these have been removed from the study design. You do need to know them in year 12 though. They are really important. So I guess there's no harm in learning about them, but just be aware that you shouldn't be tested on them in such detail as you may have been previously based on the previous study design. Okay, hopefully that makes sense. So nucleus, really important. We're thinking about the house of our DNA, so our genetic information. Ribosomes, we're thinking really important for protein production. Again, you'll learn about the really nitty-gritty details of protein production a little bit next year. So our endoplasmic reticulums, we're kind of thinking here like packaging and transport. So you can see here, it's got like modification as well of our proteins. So think about that. Again, rather than rote memorizing, rough endoplasmic reticulum does this, smooth endoplasmic reticulum does this. Think about like, what's the difference between the rough and the smooth? The rough has ribosomes. What do ribosomes do? Protein. So therefore, what does the rough endoplasmic reticulum do? Packaging, modifying proteins. You can see that kind of train of thought rather than thinking like everything separately, thinking like ribosome, proteins, smooth endoplasmic reticulum, steroid molecules, rough endoplasmic reticulum, proteins. Like think about it, I guess like think smarter, not harder, maybe like try to tie concepts together, tie organelles together. And then like obviously what I've done is a little bit simple in terms of just thinking rough with ribosomes, but that kind of thinking or logic can be applied to so many other concepts in Bio 1 too. Okay, so with that, smooth endoplasmic reticulum, again, smooth because no ribosomes, therefore not associated with our protein transport, instead with our steroid molecules. So you don't really talk about the smooth endoplasmic reticulum a lot. Like that's kind of just enough to know that it's just like synthesis of what you might call like your lipids. So just your steroids and other stuff. You like never talk about it, really. I don't think I ever wrote anything about the smooth endoplasmic reticulum in Bio 3, 4 or 1, 2. But yeah, so just know it generally. Golgi, again, we're thinking of protein. So it's basically the next step after your rough ER. So thinking about further modification, and this is more so like the actual movement and packaging of the protein ready to be sent out of the cell. Lysosomes are thinking of basically our like rubbish bins. So your unwanted stuff, your useless stuff, I guess, in the cell gets digested there. And then again, we've just discussed our mitochondria and chloroplast as well. Again, there are more organelles that may be in your textbooks and you may learn about in class that aren't covered here, but these are just some of the general important ones. Okay, so zooming in a little bit on, here's mitochondria, here's our chloroplast. Bear in mind this diagram is overly detailed. You don't need to know about like porin, we never talk about that. But just generally, again, know what it looks like. Maybe be aware of the different parts. So something important with mitochondria and chloroplast is they are double membraned, as you can see here. So they've got two membranes. And so you can see this is the outer membrane. And like obviously this is quite clear where the two membranes are. This squiggly thing is the inner membrane. So just be aware of that. Don't think of it as like something plopped in a shell. Like this is one membrane and all this squiggly stuff is actually the other membrane. So that's something important to remember. They are double membraned. Maybe remember some of the terms. Again, in terms of the locations, it's important for the process of cellular respiration. Same with chloroplast for photosynthesis, but we don't talk about that in detail a lot now. Next year, it's something that becomes a bit more relevant. But basically important terms to remember should be matrix, and cristae, really. So the cristae is this inner membrane. It's just kind of another name for these folds especially. And then the matrix is just the fluid that kind of flows inside here. Okay, in terms of the chloroplast, we're thinking about this obviously with photosynthesis, so in plants. So we have these kind of stacks. Again, I'm not going to go into a lot of detail about what they do, but we can see they always compare them to pancakes. They look like a stack of pancakes. So there's like a bunch of different names for them, but basically I just call them like thylakoid discs. A bunch of thylakoid discs together is a granum. So 1 granum, 2 grana. So again, it's like, you'll get the hang of it with the names and all that. But yeah, so think of the grana and basically the stroma. So the stroma is kind of the chloroplast version of the matrix. So the matrix is just the fluid. That's kind of the same thing. The stroma is just the fluid. You can almost think about it as the organelle version of the like cytosol, I suppose, to kind of draw a bit of comparison. But yeah, again, the names of these, the reason why I'm flagging these is because they are important for different stages. So a certain stage happens in the stroma, a certain stage happens in the grana, certain stage of cellular respiration happens in the cristae, the matrix, etc. So hopefully that kind of helps you out. Again, when you, if you like download these slides and look at them, just be aware that they are fairly detailed, like lipid globule, starch granule, like you don't need to know that stuff. Just be aware of the kind of key things that I've mentioned. Okay, a Slido poll on our organelles now. So what organelle is not involved in protein production / transport? So we've just got a couple of polls kind of scattered throughout the lecture. They're hopefully not too intense in terms of content to remember. So what organelle is not involved in protein production? Looking good so far. I think there is a little bit of a lag between what I'm saying and then once it reaches your devices. So I'll give it like 5 more seconds. Okay, looking pretty good. Perfect. I might stop that there. So the answer is the smooth endoplasmic reticulum. So with our ribosome, we're thinking of our protein production, so where the protein is actually made. Then moves on to our rough endoplasmic reticulum for further modification and transport, and then into our Golgi body, where we've got further packaging there. So remember, the smooth endoplasmic reticulum is more so associated with our steroid molecules or our lipids, kind of things like that. Okay, so moving on to a little bit of stuff more so in the cell. This is probably the most chemistry-ish it gets, or like scientific, I mean, obviously bio is a science, but you know what I mean, like that sort of stuff. So we think of things being hydrophilic or hydrophobic. So again, these would be key glossary words. So philic means to like, hydro refers to water, phobic kind of means dislike. I think of it as like, phobic, afraid. So just think of that, like hydrophilic, like kind of going towards water, liking water, hydrophobic, almost like repelling water. So that's essentially what we think. If something is hydrophilic, it likes water, it will dissolve in water. So you can kind of think of sugar. When you stir sugar in water, that dissolves. So you'll also find that we refer to that as polar. If you do Chem 1-2 or maybe Chem 3-4, I don't know if you're doing a 3-4, this will be a really common word that comes up. Lipophobic, yeah, I never really use these terms. I would either use hydrophilic or polar. And then so the opposite of that is hydrophobic, so repelling water. So again, think of like when you stir sugar in water, that dissolves. When you stir oil in water, that separates. So hydrophobic repels water. And so we can call that non-polar. And the thing here is that hydrophilic or polar molecules will mix with polar molecules. And hydrophobic or non-polar molecules, as we see here, they mix with their other hydrophobic molecules. So they're very separate. They only like to mix with the same type of molecules, if that sort of makes sense. So yeah, here's kind of a visual depiction of what we were saying. No matter how much you try to force them to interact, there's that separation. Whereas if this was water with a hydrophilic molecule, you'd be able to shake it up and then you wouldn't see a separation because it would be mixed in. Okay, the reason why we talk about this kind of stuff is because we look at it in the context of the plasma membrane. So you can see this is meant to be a cell. So the plasma membrane, what we were talking about before, in terms of how, like chloroplast and mitochondria have a double membrane. So the plasma membrane just refers to the membrane that wraps in the cell, essentially. So it looks like this. So you can see that it is 2 layers, which we'll talk about. And we often refer to it as this fluid mosaic model. Again, that's something that may come up. I probably won't go into it in detail here, but it's just this idea that the plasma membrane allows stuff to pass through it. And it's also got, as you can see here, it's quite complex. It's got other bits in it as well. So we talk about the plasma membrane in the context of transport. So we can see here, as I mentioned, the 2 layers we've got. So there are phospholipids. Don't really worry about the word like amphipathic. I never used it. Just think about this idea that they have both the 2 words we talked about before, hydrophilic and hydrophobic. They're the 2 components mixed into one, which I know I said wasn't a thing. So that's why one end is hydrophobic and one end is hydrophilic, as you can see here. So the head, it will mix with water. The tail does not like water. So that's why we get this kind of formation like this. The tails gravitate towards each other and they're on the inside of the plasma membrane, and the heads will face the outside because on the outside will be water, will be polar molecules, right? So then that's why the phospholipids basically orient themselves in this way. And then by doing that, they form this plasma membrane essentially. So that's why you'll often hear the phrase like double layer phospholipid wall or something like that. Or here, phospholipid bilayer is probably a nicer way to put it. And so that's what basically makes up the plasma membrane. So for a molecule to pass through a plasma membrane, so basically from inside the cell to outside, or the reverse way, they have to get through this phospholipid bilayer. That's just a bit more of an in detail drawing of that. So in terms of things that are embedded throughout this plasma membrane, we have cholesterol. So I suppose like these little yellow things are resembling our cholesterol. So these are important basically in maintaining fluidity. So they don't allow these molecules to get too close, but they also don't allow them to get too far. That is the general principle of it. So it's really important in making sure that essentially the plasma membrane is fluid enough, but not too loose. Hopefully that kind of makes sense. Also got this idea, of forming a glue. So you can see here, preventing them from becoming too rigid, preventing them from coming too loose. Okay, so here is where I guess like the money is. We're thinking about transport across the plasma membrane or across membranes in general. So the most simple kind of form of transport we can think about is diffusion. So this is basically when there's no like airs and graces about it really. You just have molecules coming from a region of like a high concentration and they just move to a low concentration. You'll often describe transport using these terms. So high concentration, low concentration, passive movement, net movement. And for me, I think like it felt like I was learning something foreign, but if you just break it down, like think, when you see here, like you've got something in a kind of clustered environment, it's just natural that it wants to spread out. So remember with diffusion, you don't necessarily have to think of it in the context of cells. Like if you think of perfume, when you spray perfume, when it comes out of the bottle, it's in that high concentration. The natural inclination is to diffuse. So it wants to reach out into like other corners of the room, where there's a low concentration of that perfume, if that sort of makes sense. So hopefully you can kind of understand it that way. And you can see with the diagram, it starts off, I know this is like really slow moving, but yeah, it starts off in this cluster and it's not the natural inclination for the molecules to stay there. They want to separate. And the idea is they're going to spread out equally. So it's not that you're getting a cluster from here moving, like all of them moving to the bottom right corner. they're spreading out equally. It's that idea of high concentration to low concentration to achieve this hopefully like equal spread of molecules. When we refer to the word passive, it means it doesn't require energy. If you have something going down a concentration gradient, be familiar with these terms as well. Down a concentration gradient versus against. Think if you're going down, you're going from high to low, down a concentration gradient. That doesn't require energy because, again, it's the natural pathway that the molecules will take. If you're wanting to move molecules from low to high, that's unnatural. That's going to require energy. But we'll talk about that a bit later. But that's just what passive means. It doesn't require energy. It's just what naturally happens, really. Okay, so we can see that there. Okay, so we also have facilitated diffusion. So when we refer to simple diffusion, it's basically what you think of when you think of like general diffusion. So in terms of the plasma membrane, it's molecules moving straight across that lipid bilayer. Remember, sorry to flip back and forth. The inside of the bilayer is hydrophobic. Yeah, the tails, these repel water, whereas the outside is hydrophilic. So just keep that in mind as we go through transport. So we can see here things that travel by simple diffusion, hydrophobic substances. So this makes a lot of sense. You may think like, their heads are hydrophilic. How do they get through? It's like the heads are only like kind of little, so they can just like weave their way through, really. But if you're thinking about like alcohol, steroids, these hydrophobic substances, they remember the tails. So that hydrophobic tail portion, they want to interact with them because they're the same. They're hydrophobic. So that's why they just scoot straight on through. You can have some molecules that may not necessarily be hydrophobic, such as water, but because they are very small, they can just squeeze through as well. Even though the tails like don't really like them, they can get through. And remember this idea of them being uncharged. So they're not positive molecules, they're not negative molecules, so they can still get through. In terms of facilitated diffusion, again, this is diffusion because it follows this general definition of it's passive, there's no energy required, and you're going from high to low, but it's facilitated because it requires a transport protein. So if you remember that image we looked at before, they have, sometimes these protein channels, and they'll basically be like a little gateway to go through the bilayer without actually having to cross those phospholipid tails, because obviously they wouldn't allow them to. So by having these transport proteins, it allows our big molecules, our hydrophilic molecules, and our charged molecules, so ones that wouldn't be able to go through without the transport protein, it just allows them to scoot through, basically gives them a direct channel. So that's what we'd call facilitated diffusion. Again, it makes sense in the name. Simple diffusion, you just go through. Facilitated, you need something to facilitate the diffusion. Okay, the other one we look at is active transport. So this is what I was talking about, kind of the opposite of diffusion. We're going against that concentration gradient. So we're going from an area of low concentration to an area of high concentration. So because we're going against that concentration gradient, we need ATP. So basically you need this energy to like shove the molecule against what it naturally wants to do, if that's what it makes sense. You can kind of think of it as like, when you think of down a concentration gradient and against a concentration gradient, think of against, you're going up a hill, you need that energy to kind of push things. Whereas if you're just going down a concentration gradient, you roll the ball, it goes down. You don't need to, constantly push it down the hill. That's kind of how I used to think about it. But yeah, so you can just see this here from a low region to a high region. Okay, in terms of osmosis, this is a very specific definition you have to be aware of. So net movement, you would have seen the word net in the other definitions as well. Basically, because if you remember, you can kind of see it in here, but like that little GIF, that little animated thing that we looked at with the red molecules dispersing through the black square, the molecules never stop moving. So they reach what we call equilibrium. We saw think about that perfume, like it'll get everywhere. And it's not like the molecules, once they've reached, equilibrium and once they're nice and separated, they don't just freeze. Molecules are always moving. Yeah, think about your general, physics principles. They're always moving. They never stop. So that's why it's called net movement, because ultimately once you've reached that equilibrium, you will have some molecules that go back, if that makes sense, like back to that direction of high concentration. But obviously, if you have one molecule moving that way and 70 moving the other, it doesn't really make that much of a difference. So that's why it's net movement. So net just means overall. So hopefully that makes sense. So if we can see here, I'll explain osmosis in a second, but you can see that the movement is generally to the right. Some molecules will be moving to the left because that's just how they move. But again, it doesn't really matter because it's the net movement that we're looking at, the overall. Okay, so net movement of water molecules. So when we're talking about osmosis, specifically talking about water, so osmo, yeah, it means water. The other ones we're looking at, active transport, diffusion. Again, I could be talking about perfume. It doesn't really matter. With osmosis, we're talking specifically about water, and we're talking specifically across a semi-permeable membrane, and it's from a region of high concentration to low concentration. So with the other ones, again, like simple diffusion, in this context, we're thinking of it against a membrane, but it doesn't necessarily have to be. With osmosis, we are thinking of that semi-permeable membrane. So we think about the plasma membrane, basically. That's kind of what they're referring to here. So we're going from a region of high water concentration. So if you look at the left, there are more water molecules there. Like you can like count them individually. There are more water molecules on the left than there are on the right. So the water is going to want to achieve equilibrium, and that's why we're getting water moving to the right. Hopefully that kind of makes sense. And that's why we see the water level changing. So the amount of water in the left decreases and the amount of water in the right increases because we're moving towards that right side to try to equal the level of water, if that's what makes sense. So hopefully that is kind of okay. Okay, so in terms of osmosis and what direction things go, so you have your hypertonic, your isotonic, your hypertonic. So for this diagram, this is inside. So obviously this is your plasma membrane and this is outside. So if you were to describe a solution as hypertonic, basically there's more solute. So you think of again, think about the word. So tonicity is basically, again, I don't want to get too technical here. But tonicity, basically you can think of it as like how much solute is there. So in this case, we think of sugar. So you can describe sugar as like solute, basically. It's just something that's dissolved in water. So with our tonicity here, it's hypertonic because there's more solute in this area than here. So the outside of the cell is hypertonic compared to the inside, hyper, more, yeah? So what water is going to do is, if there's more solute outside, that means there's less water outside, right? So water is going to move from inside to outside of the cell. It's going to move to the hypertonic solution. And that's what we kind of see here. So this is hypertonic. Water is going to go to it to try to balance out the amount of water, right? Or to balance out the concentration. If something is isotonic, they're pretty equal. So outside, there's kind of the same amount of solute as inside. Remember, water will still move, but the net movement will kind of just be the same. So the amount of water going out is the same going in. Hypotonic, you're probably seeing where I'm going with this. It's the opposite. So there's more solute inside, or as I should describe it, hypo, lower. There's a lower amount of solute outside than there is inside the cell. Therefore, we're going to see water come in. What the application, I guess, for this is, if you place a cell, so we're thinking like this little yellow cell here, if you place it in a hypertonic solution like this, water is going to leave the cell and the cell is going to shrink. If you put it in a hypotonic solution, water is going to come into the cell and the cell is going to swell. So that's kind of the general application. Hopefully that makes sense. Okay, so this, I'm going to go through this fairly quickly, but it's about surface area to volume ratio. I know it looks like a lot of maths. You can kind of break it down when you have a look at the slides. But the reason why you talk about this is because the question you're meant to ask yourself is, why are cells so small? And the reason is the smaller the cell is, basically the higher the surface area to volume ratio is. So you can see with the bigger cube, you have a 2-to-1 ratio. So there's 2 times as much surface area, so the outside, as there is like what's inside. It gets a little bit smaller. Now you have 3 times as much surface area compared to what's inside. Smaller again, you have 6 times as much surface area compared to what's inside. So the smaller a cell is, the higher the surface area to volume ratio is. And that's beneficial because think about the plasma membrane. Think about things coming in and out. You need nutrients, you need oxygen, you need stuff that's important to come in, and you need waste to come out. So basically the cell has to be very, very efficient at transport. It needs things in, it needs things out, and it needs it done properly, and it needs it done on time, right? So that's why cells are so small, because it means they have a huge surface area compared to the volume of the cell, so that the exchange of, nutrients, the exchange of molecules is maximized. I think a way my teacher used to say is, if you can think about like the nucleus or something, or think about a dot in the center of these cubes, think about how long it would take to get to the center of this cube, right? Based on the size. And you can see that the smaller you get, it's easier to get in because there's more surface area to that volume. Hopefully that kind of makes sense. I feel like this is something that may take a while to sort of grasp, but hopefully the general principle is kind of there. Again, be aware, I am going through these things fairly quickly. You will learn them again in class, obviously. And so don't expect, like, if you're not grasping it immediately, don't think like, oh, I'm so silly. Like, it takes time. So don't stress too much.